Hello friends, welcome to day 2 of week 7. So in the previous weeks we have looked at various assembly methods and also like methods to do the hybrid genome assembly, which we'll also like do a quick revision today. So basically we have understood the process in which we generate the data, we correct the errors, we take the corrected reads for assembly using either the short read based assembly, long read assembly, or even the hybrid assemblies, right? So, but still even after the hybrid assembly there could be gaps which may remain, and you would like to close those gaps, and also you would like to correct and polish those assemblies so that you can remove or reduce the errors as much as possible, right? So for this the data may be available with you either in the short read and long read, which you can use further to refine the gaps, because initially your aim was to assemble it, but now your aim is to clean the assembly that you have made and also reduce the gaps or fill the gaps, right? So we will see some method that we commonly use and which are important and standard methods for removing the gaps and also preparing a better finished version with lesser errors. Okay, and once you have made this assembly, then the next task is like, how would you know that the assembly that you have made is good quality assembly, whether it is complete or not, right? And in what statistics you can express it so that anybody you like who looks at this assembly can get an idea of like what is the assembly quality. Okay, so we'll cover these things in today's class. So basically we are generating data either by the short reads, that is second generation NGS technologies, including the Illumina or iTorrent, right? And that produces like short reads of like 75 to 300 base pairs, and then commonly 150 base pair reads you will get, and the long reads that you get from either ONT or PacBio, right? And you use those reads because they have their own benefits and limitations, and combination of the two basically leads to a better quality assembly. So, and later on once you have done this, you will go for the chromatin assembly and that means like chromosome level assembly, that how many chromosomes you can find, and can you basically segregate the contigs or scaffold that you have into various chromosomes. Okay, so that is the next task. So using both the assemblies we can cover the gaps, we can improve the quality, right? But still there would remain like some gaps because either because of repetitive sequences which are difficult to like place in a genomic context, that which place they may fit, because it may fit at multiple places and also overlap with multiple regions, right? So this is one challenge. And also like the presence of like genic regions and intergenic regions and regulatory regions also poses challenges, and also this genic region can also be used to actually assess the quality of the genome, that we will see today. Okay, so basically short read assembly is done by like so many tools are there, but basically they use the De Bruijn graph based approach, where basically the reads are broken into small k-mers of of various lengths. That length also you have to optimize, or you can do iteratively and look at what is the best length for the assembly, and then basically it looks at the common it builds a graph based on the overlapping k-mers, right? And using that graph it builds the consensus sequence, which is like here. Okay, so this is De Bruijn graph. So many tools are available which are based on this, and this is one of the standard approach for the short read assembly. And for the long read we basically rely upon the overlap layout and consensus approach, where first in the long reads, okay, basically in this one the k-mer approach may not work because we don't want to break it into smaller pieces because it's already assembled and long pieces are there, right? So what we do is we look at the overlaps between the reads, that is the overlap, and then based on the overlapping reads which are above a threshold, we lay the reads according to their overlap. So that is the layout of the reads, and then we build the consensus, that is arranging the reads which are overlapping and building the consensus. So overlap layout consensus is the basic algorithm which is used by several programs which deal with the long read assembly, either PacBio or ONT. Okay, so short reads are more accurate, and but they are short, so difficult to assemble, right? And they may lose the information about repeats, and those regions are difficult to assemble by short reads. Long reads are good for high contiguity, and but they suffer from their error rate, which is improving, but still it is we require the short reads for this. Okay, and the cost, like short read may be less expensive, long reads more expensive to produce, right? And like these are the various short read assemblers that are available. Like most common one is like the Spades, which is like have so many citations. 18,000 people have used it and cited in their papers, and yearly also like more than 2,000 or almost 2,000 publications, followed by several other previously published tools. Okay, and this is perhaps one of the latest one, right, which is now used, right, and all have their own advantages, and but Spades generally does well for the short read assembly. Okay, so basically overall schematic chart looks like this. Basically you find the genome, do the sequencing, produce the short read or long read by the sequencing, right, and either of the instrument Illumina or Chromium, that is link reads, and the short reads and long reads by PacBio or Nanopore. And then basically long and short read together as using hybrid, where either DBG to OLC, De Bruijn graph to overlap layer consensus, Masuka or ONT assembly method is there. Or you basically, for if you are dealing with the link reads, you will be using Supernova that we discussed in the previous lecture, right? And for the short reads, ABS Velvet or Spades are the tools, right? For the long reads, Canu, Fly are one of the some of the common tools for this. Okay, so basically hybrid approach is needed because we are dealing with genomes which could be very large in size. They may be present in they may be polyploid, so multiple copies of the genome could be there, right? So okay, polyploidy increases the complexity, and it is also difficult to resolve the haplotypes in case of polyploidy. Then heterozygosity, which is an important issue, because if it is more than 1%, then basically resolving the k-mers or basically arranging the k-mers using De Bruijn approach, or okay, so that becomes a difficult task, right, by shorter reads. And the presence of repeats which are there, also like in plants it could be almost 80-90% also, sometimes repeats are there. So you have to resolve these issues, and that is why hybrid assembly is required, and we have also discussed it in the previous classes. And basically once you have the hybrid, so there are different ways of achieving the hybrid assembly. Either you use the short read data, okay, to improve the long read assembly, or long read data to improve the short read assembly, right? Or you create an hybrid assembly and then use both long and short read data to improve the assembly. Okay, so basically either one, either like the data from the third generation sequencing technology and second generation sequencing technology are used to generate the hybrid assembly, or either you use the SGST short second generation data to correct the errors in third generation, or basically the other way, like using the third generation to improve the contiguity in the second generation. So different approaches are there. Okay, but generally like once you have made the assembly, and basically since third generation or longer reads are better because they can cover large portions including complete genes and repetitive regions, right? So correcting errors in those reads is a better idea, and that is why the second generation sequencing technologies or data can be used there. Okay, so today like we will be focusing on these parts, that the gaps that remain and how to correct those gaps, and also there could be some errors in the assembly, right, which were not taken care by the previous assembly methods, and how we can correct those errors. Okay, so basically in the hybrid assembly, as I mentioned, there are like 2-3 ways. One is like either short reads are used to correct the long read or the hybrid assembly, or long reads are used to build the consensus, and there are some pipelines also which are available. So we have dealt with these things in the previous classes in detail, what is hybrid assembly and how we do and what are the different tools. Okay, two tools basically we use commonly, quick merge and ragtag, and in just one slide I will tell like just to revise what those tools are and what were their basis. Okay, so basically the idea is to use the short read data. So basically you have short read data and long read data. So you assemble the short read data, okay, and once you have done the scaffolding, at this place you can use the long read data for scaffolding, improve scaffolding, because they can place short reads in a particular order, right? Even if it could be like disjointed contigs in case of short reads, but they can be placed in reference to longer reads while doing the assembly scaffolding. Or you can have long read data, you can generate the assembly and then polish this using the short reads. Okay, and then basically once you have achieved an assembly, again you can go for another round of gap closing and polishing. So we will be dealing with this and these steps in today's class. Okay, so yeah, so basically again like you have reached to this point where we have already done the scaffolding, right? And now these kind of gaps and the errors that are there, we want to close, right, and improve the errors in the assembly in hand, okay, by polishing. So basically polishing means improving the a kind of draft finished assembly so that you have lesser errors and or minimize the errors as much as possible. Okay, correct those errors and also fill up the missing gaps, right? And the final genome assembly that you have, we can measure its completeness, correctness, and also like different parameters to describe about it. Okay, like how good your like sequence in hand is, we will see today. Okay, so quickly, like quick merge, what it does is basically it is finding the overlap and merging the sequence. So basically it is a way in which we can merge the short read and long read data, okay, or basically their assemblies together into a final genomic assembly. So basically to find the overlap between these, it uses mummer tool where it compares. So what it does is, for example, you have this pack bio contig or long reads, okay, or a hybrid assembly. So it will take these and find the overlap between the two, and there could be some overlaps which are more reliable, okay, like this, and some overlap which are less reliable, okay. So basically because, and that is why it will score them as highly high confidence overlap and the low places where it could not find like high confidence overlap, okay. So basically so it will be using the high confidence overlap with the longer contigs, okay, as the seed contig node and others as the contig node. So basically the idea is that those contigs that show higher confidence overlap should be connected together, right. So once it finds those contig, make the seed contig, like in this case if you see like blue, orange and this, okay, are taken here, and in this case you can see like maroon and blue color. So basically you can see they had good nice overlap, so it is connecting those, right, and finding the ones with the dotted lines means like low confidence HCO, and the complete lines means that there was a high confidence overlap was found, and based on that it connects or merges the two, okay, the contigs, and make the consensus or final assembly. Okay, so you can either accept one of the genomes as the donor or acceptor based on like if you want to use the short read data as the donor and improve the long read, or long read data as the donor and improve the short read. Although like always like or mostly you will say that using the short read as a donor works better, okay. And ragtag is also like a tool to basically build the consensus using the long read and short read, okay. So what it does is basically it first of all identifies the regions in an assembly which are unfilled or basically gaps in the previous one, okay, and then it tries to fill up those gaps, which we call patching, okay, in that target assembly, from the query, right. So one of the query assembly takes as query and one as target, okay. And then it also tries to merge the two so that it can complete or basically merge the assemblies to make it more contiguous, okay. For example, here you can see that there were gaps, there were unknown base pairs, okay. So basically it used the information from the query to fill up these parts and made the merged or kind of overlapped assembly, right. So this is also a common method. Okay, so now what we want to focus is like how we can close the gaps that may still remain after doing the hybrid assembly, right. And also there could be errors in the hybrid assembly because you are using both long read and short read, and long reads are prone to errors, and you may have used the long read as the template and basically short reads to fill up those gaps, okay. So long read can still be corrected, or basically the draft assembled assembly can still be corrected by using the information that you already have in place in the form of short reads or long reads, right. So basically we have to in today's class we will be dealing with like what are these gaps and how we can fill up these gaps, okay. And also there could be errors in the assembly which we want to correct, okay. And basically the gap closing or these error methods use the same. If you are using short reads, it will be using de Bruijn graph. If you are using long read, it will be using the OLC method. So basically those algorithms are also based on like sealer uses the DBG approach, and the gap closer uses the TGS gap closer uses the OLC approach, okay. So TGS gap closer is one of the method that is used to close the gaps in the assembly, okay. So closing the gaps means that okay, there is a gap at this place, okay, but we do not know that these two contigs can still be connected, okay, first, right. And or it could be a scaffold where we know this and there could be ends present over here. So you want to fill this information, and also there could be errors here and there which you want to correct, that we can generally do with pylon, okay. So what it does is, first of all, it uses the draft assembly that you have already created, okay. It takes the raw long reads and raw short reads, okay. So all three things you already have because you are dealing with hybrid assembly. So you had the short read data, long read data, and the assembly, draft assembly. Then it identifies that what are the gap regions like this, okay, and find the appropriate piece that can fit in that gap, okay. So basically it finds the appropriate piece that can go into that gap and fill this gap, okay. Now that appropriate piece may contain sub errors, okay. So it does the error correction also, okay, in that piece first using the short read data, okay. So if I map the short read on this part, okay, so basically if there were some errors here and there, I can see that what bases are present in the same location in the short reads, and I can get more confidence about that base pair or accept or reject that erroneous base pair. So it does the error correction in that read, which in the piece that should fit a gap, okay, and then fit that piece in the gap and update the assembly, okay. So basically in detail if you see, so basically taking the draft assembly or long reads only, okay, and then it will map the long reads here in the first step to fill up the gap, because to fill up the gaps basically long reads will be more useful as compared to the short read. And in the long reads basically you might have chosen a cutoff also during the initial assembly or only the long read assembly, that okay, reads above 5 KB should be taken, right. But there were reads which are just like less than 5 KB, that means 1 KB, 2 KB, 3 KB, and intermediate ranges reads, okay, that were not used in the assembly process but still can be used to fill up these gaps, okay. So the raw long reads from ONT or Packboy are used, okay, and mapped on the draft assembly to find the fragments which can fill up the gaps, right, okay. So so basically we know that some gaps have to be filled up, right, and so we will find the long reads that align well into those regions. Okay, and then on those reads we will map the short reads. Okay, so in this candidate fragment that we have got, we will map the short reads. Okay, and correct the errors in this read using the information from the short reads, right? And after the error is corrected, we will the corrected candidates we will take and use that as new candidates to fill up the gaps. Okay, and then the new candidates that we have got, we will again map it on the genome. Okay, and then there could be multiple new candidates also. We will sort them according to the best match. So if they are overlapping and basically they are with the region that we of our interest or the gap, okay, we will select those. Then we will see that whether they are passing our filters of the threshold that we have kept. If yes, we will use that for finishing the gap. If they are not overlapping, basically we will be rejecting, of course. Okay, and if they are not passing the filter, again they will go back and maybe use it at some other place and like that. Okay, so this is how the gaps are closed by the TGS gap closer. So this is a tool which is available okay, at this place. Basically GitHub is a site that houses most of the bioinformatics software, and basically the developers or the authors are also encouraged to submit their software at GitHub so that others can use it and download it for free from there. Okay, and this article was published in Giga Science. Then next step is to assembly polishing. That means correcting of errors that may be present in the assembly. Okay, so Pylon is one of the tool to do this. So like this is your final assembly, but there could be errors in this, right, at various places. So what you will do is you will map the short read data, okay, using bowtie or BWA on this long read. Okay, short reads map to long reads. So now for this particular position, you can see that what is the base present in the short reads. Okay, so basically there could be A in your read, and it could be A or C and A. So basically you can see that most of the short reads are making a call of A, so you can fix the call as A, right? So this way you can correct the most of the errors which are there in your long reads, or basically your final assembly, right? So Pylon is one of the tool that basically aligns the short reads on the final assembly and correct the errors. Okay, so like this. So basically it looks for this was your long read, and so it looks for the error and fix up the errors looking at the information from the other short reads. There could be also be like some local misassembly. For example, in the assembly you are this is a region, whereas in the short read basically information is coming that basically this region should be flipped, or slightly the assembly has been misplaced, so it will also do that correction. Okay, so basically it may reassemble the and close the gaps, and across the gaps basically generally these errors happen, and so it is fixing the errors and also kind of rearranging a little bit so that you can fix up those assembly errors. So Pylon is a good tool, okay, which is used for this purpose, and it's a commonly used tool basically to correct the errors in the final genome assembly. Okay, and it was published in PLOS One and available again at GitHub, which is the source for all the free bioinformatics software that are published. Okay, another approaches are also available, such as SeqBug, which was used to correct errors in the tiger genome. So basically the idea is same, that there already exists an assembly, right, but you think that that assembly has errors because you are looking at doing the analysis, but you are finding the genes you are getting from that assembly have premature stop codons or basically truncated CDS, okay, because of stop codons in the CDS. So that means there could be errors in the initial assembly because some of these stop codons or these may be acceptable because they could be a result of evolution, but if they are finding it too many, then basically there could be reasons to believe that or basically looking at the similar or closely related species, you can see that okay, if they did not have this error, so why this species is having, and you may go for one round of correction of that using resequencing. Okay, so the idea is that for any given region, you will again align the reads to that region and look at that what is the consensus or the base pair in those regions. So basically it was A here, so you can see that at position 1 it was like A, C, T, G. So mostly it was C in that position, okay, but original base that you have called, which was only occurring 2 times out of 42 occurrences at that position, okay. So basically that base should be replaced by, so basically what was happening here is there is one consensus, okay, where the base was T, but in the reads that you have mapped, mainly the base was C, right, and only in like few cases it was T, that was like just 2 out of right, all the 42 reads that you could find that were matching to this region, right. So that is what that means, that by mistake T was picked, which was present in one of the reads, but now it should be corrected to C, and this way you can correct the errors in the assembly, okay. So basically reads have to be mapped on the assembly and aligned well, and based on the alignment you will calculate that whether there was error or not, okay. So so basically again you have to take care of like what is the coverage of that. That means at least 10 reads or 10 more reads should be present which may align to that region. So basically this is your consensus, and there should be 10 more reads, okay, which are at least there, and at least you can say like 1/5 of the reads should not contain that base, otherwise it will be confusing. So you have to give some threshold that, okay, if it is like A 50% time and G is 50% time, then you don't know whether it is A or G here, but like 8 out of 10 times it is coming A, then basically it is more reason to put A at that place. So these algorithms are based on such kind of logic. Okay, so basically for example this was used for the resequencing of tiger. So tiger genome was there, but authors observed that it had several errors in the genic regions, like 11,000 errors in the genic region, and of course in the genome like almost 9 lakh or basically 1 million errors. So of which like 11,000 were in the coding region, which is a huge number because it will map to like almost 6,000 transcripts and 4,000 genes. So 4,000 genes contained RNS base, and if that RNS base is taken, so it may lead to false positives when you do the evolution analysis. You may think that, okay, these base pairs or these amino acids are changed in these genes, right, and you may think that it's a sign of evolution, but it could be the error that you have done during the assembly process. So correcting these errors and generating a high quality assembly, looking for the errors, correcting the errors, polishing the assembly, filling the gaps is very, very important process, right, and that basically leads to a better assembly which is more publishable, and the analysis that you do from that will also be more reliable and will stand the test of time in the future. Okay, so basically the process is take the data, do the shorted and long read assembly, okay, and after that merge the two assemblies, polish the assemblies using one of the tools, okay, and either long read and shorted or both, right, depending upon your way of doing, and you may adopt any of the method, doing correcting with long read and shorted both, or just one of those, using shorted preferably. So this way like you can do. Now the assembly is there. Now you have to report some statistics from the assembly so that others can know that how good is your assembly, okay. So one of the statistics is N50. So this word we have called several times in the previous lectures also, and you will be hearing whenever you are talking about a genome, what is the N50 of the genome. This is the first thing that you'll ask, okay, and that tells that basically how big the contigs are in the genome and how big is the contribution of large contigs in the assembly that you have, okay. So basically it is the median statistics, that means 50% of the entire assembly should be represented by the contigs, okay, which are equal or larger than this value. Okay, for example, so basically the idea is to see the number of contigs, okay, and the median contig which basically represent at least 50% of the assembly. So to make it like more clear with an example, like there were, say for example, 9 contigs in an assembly of length 2 kb, 3 kb, 4 kb, 5 kb, 6 kb, 7 kb, 8 kb, 9 and 10 kb, and together they are making the total sum of 54 kb, okay, right, if I add all these together, right. And basically these this genome size was also 54 kb. That means you have kind of covered all the genome in the contigs that you have in hand, but still you have contigs and not the proper assembly or contiguous assembly. So you should look at like how good is your assembly. So basically what is the half of this number 54, which is the genome size? So basically that is 27, right. So that means half of that sum of all the contigs, the half of that is like 27, 54 by 2, okay. And now we will see that, okay, if I look at half of that value 27, how many contigs represent that? So basically if I sum the 10 plus 9 plus 8, basically that equals 27. That means at this point I can say that these 3 will contribute to 50% of the genome, okay, or the total assembly that I have done. So this will be my N50, that is 8 in this case, right. And basically rest of the assembly on the left side represent the shorter contigs and the right side part represents the longer contigs, okay. So basically N50 in this case is 8, okay, right. Now the second thing that you should look at here is that L50, okay. Then okay, N50 is okay that it is 8, and because like 8, 9, 10 and like these 3 contig and like half of that the total assembled genome is is kind of median that separates the two sides, okay. Now in how many pieces is the assembly that you are talking about, the N50, okay. So basically it contains 3 contigs 8, 9, 10, okay, 3 pieces, okay. So your L50 will become 3. So N50 tells that what is the biggest contig or the largest contig that is kind of median to the total assembly size, okay. Okay, so that will tell that bigger this value, better is the assembly, okay. Then L50 tells that how many contigs or fragments actually represent your 50% of the assembly, okay. So basically lower the number, better it is. So for example, if you have just 1 here, that means you have got single piece the complete chromosome. If it is 3, that means your chromosome is in 3 parts, okay. There are 3 contigs that represent, okay, this part, okay. So and basically like N50, it's N90 is also there, like if you are dealing with 90% of the assembly, that how many what is the N90 value and how many contigs represent that value. So it is even more stringent, okay. So like taking another example, like this is your 2 million base pair genome, 2 MB genome. So if these are the different length of contigs here, and basically you can see that there will be very long contigs in the genome, there could be very short contigs also, because you have done random assembly and the pieces that could find an overlap and merge, and basically you could build a contiguous assembly there, okay, became contigs, and there could be short contigs and very long contigs also. Even the large and small reads, long and short reads play a role here. So if you see this picture, so basically halfway if I say, basically it is somewhere here. So I can say that, okay, based on the total assembly size, okay, which was say this number, okay, half, like up to 100, basically I am getting a median point where it is representing at least half of my assembly, okay. So basically N50 in this case will be 100 kb, right, and greater this value, because if I had got a value of 200, that then I would be like having only 2 pieces of the total contigs or scaffolds that you have, okay, and that means a better quality assembly. So lower higher this value N50 is a better idea. And similarly, like in case of L50, the L50 will be how much? It will be 4 here, 1, 2, 3 and 4. Okay, so another important, so this is basically N50L50 tells more about how good your assembly in terms of contiguity and number of pieces. But now you should also see that whether your assembly is complete or not. Okay, number wise counting those contigs, you may say that okay, this is actually exceeding by a number of the base pairs in the genome, or you have covered the entire genome. Okay, but whether you have really covered that genome can be expressed in terms of a BUSCO value or BUSCO score, okay, which is basically benchmarking universal single copy orthologs. So single copy orthologs which are derived from various branches of phylogenetic tree, or you can say the different clades, including vertebrates, animals, plants, eukaryotes, even bacteria, like prokaryotes also there, right? So what they have done is they have built a database called OrthoDB containing single copy orthologs which are present across genomes, and it is not that like all the orthologs will be present in all the genomes, but at least most of the genomes or closely related genomes should have those orthologs. Okay, so basically several hundreds of genomes with single copy orthologs were identified. Okay, and they built a set of almost 3000 genes for vertebrates, 2500 genes for arthropods, 843 for metazoans, 1438 for fungi, and 429 for eukaryotes. And also they also derived the marker genes for the bacteria, because this is a statistic that you can use for either N50 or BUSCO, can be used for prokaryotes as well as eukaryote assembly. Okay, so that was also taken. So now what they have done is, like, with those orthologs, because that represent a set of genes that should be present in the genome, and they are randomly distributed across the different chromosomes. Okay, so it is not that like they will be concentrated on one place, or okay. So basically if you find the width of these genes, like they are present anywhere in the genome, right? So okay, and if all of those are present, or some of those are present, or they are present partially, so that will tell something about how good is your assembly, because if your assembly is very good, that means you should be having all the genes or these marker genes, right, or single copy orthologous genes, and if those genes are also present, like fully, not fragmented, so that will tell that assembly is of very, very good quality. Okay, so how it is done is, so basically you take a genome assembly and run the tool, okay, on this. So basically the tool will call a gene prediction program. So first of all, it will align with the consensus, that means your the orthologs that you have. Then it will call a gene prediction program, that is Augustus in this case. That will take most of the time, because it takes some time to predict the genes, right? So that will find the ORFs in your genome, right? And basically it will use a HMM based tool, HMMR3, okay, and that is because it's a machine learning based tool, it will be little faster, okay. And then it will classify the orthologs that it has found in your genome in the terms that whether complete ones were found, duplicate ones were found, fragmented were there, or they were missing orthologs, okay, right. So basically, for example, in one of the cases it got a complete, or C, basically complete BUSCO score of 74. That means 74% of the single copy orthologs were complete and present in the genome. Okay, fragmented were 12.6. That means 12.6% of the single copy orthologs were present, but they were fragmented. Okay, that means your genome still have some fragments, and it is not covering those markers well. Okay, 13% could not be found. That 13% of the single copy orthologs could not be found, which is a bad statistics. That means like a lot, or basically 13% or more of your genome is missing in your assembly, right? Okay, so basically this way you can express the BUSCO score, and we will see some examples, okay, where we are using the N50 and BUSCO, and that will give some hint about the quality of the genome. So this tool was published in Bioinformatics and journal, and it is available for download from this site, okay. And it's a good tool that is used to determine the overall completeness of the genome. And even the reviewers will ask you to report this N50, L50, BUSCO, right, another statistics also when you are reporting your genome. Okay. For example, in this genome that we have already seen the example earlier, a riverbank grape or frost grape genome. So basically using multiple type of data from PacBio, Chromium, link reads, short reads, okay, and lot of data with high depth. Basically the assembled genome size was 500 MB, and but the N50 was only 964 KB, that means less than 1 MB, okay. So which is actually not that bad, and this much N50 is good for going for the chromosome level assembly also, because anything above half a MB or basically 0.5 MB or 500 KB is good enough to go for a chromosome level assembly using the high-C technology, right. And number of pieces were 174, which is good, right. And BUSCO completeness is also 95 and above, that means like almost 95% of the genome you could cover, okay. Then in case of this western honeybee, you can see that total size of genome was 225 MB, and the N50 was 5 MB, which is a good number, and L50, that means 50% of the genome that you assembled could be expressed in only 14 pieces, which is very good, right. And so this means that it was a good assembly. In case of this fish species which was published, okay, and basically in this case the genome size was 790 MB and N50 was 1.9 MB, which is also a good number. BUSCO they did not report here. In case of this medium size China trail Melania olifera, basically you can see that the total size the genome was of something like 1.5 GB, and basically they could find 4 MB as the N50, right, which was represented in like 94 pieces. So basically this is also means a good assembly, and there are other statistics like GC content and L50 and N50 and 90 they were also reporting. Okay, so basically here also like the long reads and short reads were used, link reads, Chromium link reads were used. Okay, another fish genome which was published in 2017. So here the genome size is like 2.3 GB. BUSCO score was 90%, that means above 95% is better, but 90% is also acceptable, and but including both complete and fragmented they could achieve 94.5%, that means they could still have other single copy orthologs but in pieces or basically not complete, okay, and a good N50 in 19 MB. Aloe vera genome, which was a large genome, 16 GB in size, right. So it was kind of drought assembly. The total length was like they could assemble was 12 GB out of that, and they also predicted this as the genome size, and the largest scaffold was 4 MB, but still the N50 was low as 14 KB only, okay. In case of turmeric genome size was 1 GB, N50 was 100 KB, but 90% of genome still they could cover, 92% BUSCO was there. Banyan tree, basically N50 was it was small genome 300 MB like that, both banyan tree and people tree, okay, 500 KB and 50 they could get, and BUSCO score was good above 95%. So that means most of the single copy orthologs they could map to the genome or could find in the genome. In case of jamun, the N50 was 179 KB, but BUSCO was 98%. That means like at least gene wise they could find most of the genes, right, in this genome. In amla also the N50 was 597 KB for a 500 MB genome, and the BUSCO was 98%, which also tells that BUSCO wise and N50 wise the genome was fine, and this is like reliable and basically a reportable level of genome assembly, okay. So basically peacock genome has been like several versions are available, but you can see that in case of different scaffolds, if you see the last version had only 69, was only in 69 pieces, and it was a significant improvement from the first version that was published, and also that there was a significant increase in the N50 of the scaffold, that is from 25 kb to almost 84 MB, million base pairs, right, and number of L50 was also like decreasing, and also like more number of genes could be predicted. So basically with time it is that when we have more data from other researchers or basically multiple versions, and like you produce a draft assembly, somebody else will use that and make a better assembly. This way there will be gradual improvement in the genome that are currently existing, but the idea is that we cover new and new genomes which are important for health or for commercial purposes or for environmental purposes, and later on like they can be improved using more and more data and technologies. There is another term called LAI score, because repeats also are important okay, to determine, and basically in case of plants this is basically LAI score is mainly for plants because they have high level of LTR retroposons. Okay, so basically the idea is that how many of those you could detect. So basically the LAI score is calculated by intact retroposons that you have found, the length of that. So basically if you have found say 100 intact LTR retroposons, so you will calculate the total length of those 100 LTR retroposons, okay, divided by the total LTR retroposons that could find. So intact by total, okay, others could be fragmented, so and multiplied by 100. So this will tell that okay, what is the percent score of basically your LAI or retroposons that you have found in the genome. So basically more this value better it is, like gold will be more than 20, draft assembly will be having like 0 to 10 0 to 10 range, and a reference quality will have 10 to 20 LAI score. So this is basically reported for the assembly quality of a plant genome, okay, and this paper came in NAR, and it is also commonly used. Recently a new tool has come called WebQuast, which actually does a lot of such statistics calculations and report a lot about your genome, right. So okay, so it does the L50, N50, BUSCO and everything like that you want to report is calculate. So this table tells that how basically they have assessed their genome on a their tool on a sample genome, and they compared the publicly available methods like ABIS, Megaheight, Spades, Velvet, which are the shorted assemblers. So basically this table tells that these are the statistics that it could calculate on these genomes, okay, and it also compared the tools and also tells the capability, the statistics that you can report by using the WebQuast, okay. So in the end it is also reporting the BUSCO score, and you can see that all the tools, the BUSCO score was almost equal, although the N50 showed a lot of variation, like this one Spades had the highest N50, right, and the L50 was also lowest in Spades. That means Spades did the best among all, although in terms of BUSCO all were like almost equal. Okay, so WebQuast is one of the tools that you may like to use to do most of the statistics that you want to report from your genome. So it's a recently published tool, and this reports a lot of information, important information about your genome. And basically these kind of tables you would also like to report in your as supplementary tables or the main tables, or using this you will make figures and use that for further analysis. Okay, so in this lecture we looked at why hybrid assembly are needed, and how to correct and the errors in the hybrid assembly, the draft assembly that you have got after the hybrid assembly, and also how to improve those errors and improve the assembly quality by filling up the gaps. Okay, then we looked at the and including the method that can do this job, which are commonly used, and then we looked at the measures or parameters that evaluate the quality of assembly, right, and how these parameters are important, because that gives you a indication of how good the assembly is. Okay, So I'm sure that basically when you now see a paper or read a paper that contain these numbers and these values, so you will be able to assess the quality of an assembly very easily. Okay, so this is good for today for this class. Thank you.